I'm now in Nashville and I'm together with Ripla Arora, who actually visit Nashville, the headquarters of TBX4Life. But she's located in Michigan State and works in Michigan State University. Could you please tell a little bit more about yourself and your work?
Thank you, Anton. Hello to the community. So I am a scientist who studies developmental biology, developmental genetics by training, and my lab focuses on understanding early embryo-uterine interactions using a lot of microscopy and 3D reconstruction of structures. And we're very interested in how organ structure affects function.
Wonderful. So I know that you're also working with animals, or animal studies, right? So in particular, we were talking about development of mice models. I think that's one aspect which we would like to ask you to clarify how these animal studies help the research in general, right? So what is the connection between these animal studies and maybe clinical trials in humans, and is there something in particular what we as a community can help with these animal studies? Certainly we cannot make a mice.
Okay. Yeah, so animal studies or any model organism studies in developmental biology and genetics, it's really important because once you find out that there is a genetic link to some kind of phenotype, it's really important to study it in detail. And with lots of molecular biology and techniques in this generation, we can create mouse models that have deletions of a particular gene or point mutations or duplications of a certain gene. And once we create that kind of model in the mouse, we can study the mouse over time and we can get a lot of information. Another good thing about the mouse model, if you think about it, is a mouse's gestation is only 3 weeks, and if you think about the neonatal period of the mouse, it's about a couple of weeks. So everything that happens in humans, it's really shortened in time in the mouse, so that researchers like us can actually create the mouse and study the phenotypes in the lab condition. And so when you ask about how the community can help us so that we can improve and speed up the research, it's collecting phenotypical data from as many people as possible. For example,
by phenotypical data and phenotype, maybe you can give a little bit simpler words.
Yes.
What that means.
Yeah. So genotype is the gene that you are manipulating, and phenotype is the observable issue that might happen when
pulmonary hypertension, for example.
Yeah. So
skeletal issues.
So pulmonary hypertension, skeletal issues, or even just we were talking about this, Anton, that for example, pulmonary hypertension in adults is more prevalent in women as compared to men. And then we were starting to talk about this, that is this really true for kids as well? And we don't have enough data to support the idea either way. And I think getting that kind of information is really powerful, so that when we create a mouse model for TBX4, we actually know, does our model, how closely does it resemble the human condition, and how good is it a model to give us information about the human condition.
So essentially, actually, with the animal model, you would like to create kind of like a copy of these problems which we experience now in humans, right? So
exactly. So it is possible that when we create a mouse model, we may not copy all the human phenotypes or the conditions. But if we know that creating a mouse model gives us pulmonary hypertension, or if we see that even in mice, the female mice have more prevalence of pulmonary hypertension, then that makes it a good model system to figure out why does this happen.
Okay. So besides kind of answering those questions, my understanding is there's also very practical approaches, animal preclinical trials, right? So this kind of safety trials.
Yeah. So for example, if you were to take a drug and you thought that you found a gene, you figured out what it controls, and now you want to test if you inhibited that signaling pathway, would that rescue the phenotype, or would that provide the mouse comfort from the problem? In that scenario, the mouse is a good model system to create the genetic mutation and then to see if the medication will help. And if you can follow the mouse for a long time, which is shorter than following human trials, you can actually figure out in the long term, are there going to be any problems with the use of the drug? And once you use mice as a model system, then you can move on to bigger animals and eventually go to human clinical trials.
I understand. Good. So what do you think, so how far we are currently from being able to have that good model for TBX4, for example?
So that's a very good question you ask. Actually, 10 years ago, for my PhD, I was working with the TBX4 gene, and in 2012, this publication, when it came out, there was no idea. We had no idea that TBX4 was associated with any kind of lung development. And in that publication or research 11 years ago, we found that if you remove TBX4 gene along with one of the homologs TBX5, then the mice have smaller lungs. And so we already had sort of a model system, but then for 10 years there has not been a whole lot of mouse research, or at least there's not a lot of published research with model systems. And so what we're trying to do in the lab right now is to use different kind of methods to remove TBX4 during development of the mouse embryo, and then we can study once the mouse is born how those mutations, or when we delete the TBX4 gene, how does that affect the development of the lung in the neonatal young mouse. And I think once we have the models, which is, I imagine quite a few labs are actually working on this to try to remove the gene at different time points during development. And with the sort of advent of molecular techniques like sequencing, I think we are getting closer and closer to get this mouse model. And once we have the mouse model, that could then set the stage of a sort of model where you can test drugs once you figure out what
like
kind
a
of
drug discovery.
like a drug discovery. Yeah, so you would want to know what kind of proteins does TBX4 affect, and then you would want to try to stop them or increase them using some kind of drug manipulation, and the mouse model would be very useful for that.
I understand. Wonderful. So yeah, this is very interesting. Is there some particular message or something you would like to share with the community or wish to TBX4Life?
Yeah, so I would really like to say that I got back into the TBX4 research about 1 year ago when I met the TBX4Life community, and I think that the more we come together as a community of researchers, patients, families, clinicians, geneticists, I think the faster we will get to a cure. I mean, think about COVID. The whole world came together and we had a vaccine in a year. So I think if we all come together, we are that much closer to a cure for TBX4 syndrome.
Amazing. So thank you very much, Ripla, and thank you being part of our team and making this together.
Okay, thank you.